The discovery that somatic cells could be reprogrammed to a pluripotent state has profoundly altered the landscape in which stem cell research is conducted. Today, we will focus on the following three aspects: iPSCs, access to iPSCs, pluripotency validation. Part 1, iPSCs. iPS cells are derived from somatic cells and they are similar to S cells in morphology, proliferation and the ability to differentiate into all tissue types of the body. Human iPS cells have a distinct advantage over S cells as they exhibit key properties of S cells without the ethical dilemma of embryo destruction. The generation of patient-specific iPS cells circumvents an important roadblock to personalized regenerative medicine therapies by eliminating the potential for immune rejection of non-autologous transplanted cells. The application of iPSCs in biomedical research mainly includes basic research into pluripotency and differentiation, applied research into disease-specific model systems and translational research used in regenerative medicine. Part 2, access to iPSCs. iPS cell reprogramming was originally discovered by the overexpression of 4 transcription factors, OCT4, SOX2, KLF4, and c-MYC, later dubbed the Yamanaka factors, with a retroviral delivery system in mouse and human fibroblasts. Additional genes, however, including certain members of the KLF family, KLF1, KLF2, KLF4, and KLF5, the MYC family c-MYC, L-MYC, and N-MYC, NANOG, and Lin28, have been identified to increase the induction efficiency. Proper utilization of the technology to generate induced pluripotent stem cells requires that researchers select the appropriate reprogramming method for generating iPSCs so that the resulting iPSCs can be transitioned towards clinical applications effectively. The reprogramming methods can be divided into 4 groups based upon efficiency, footprint, and number of different somatics types known to be reprogrammed. How these factors are weighted will be dependent on the goals of the project. Projects with translational aspirations should utilize a completely footprint-free method with consideration of whether fibroblasts or blood cells will be reprogrammed. Sendai virus works well with all cell types but requires 10 passages for the generated iPSCs to be footprint-free. Reprogramming using the episomal method is excellent for blood cells but requires modification of standard culture conditions for fibroblasts. There are also differences in the amount of time it takes to lose the footprint between episomal and Sendai-based methods. There is no need to worry about the vestiges of a footprint remaining in iPSCs reprogrammed by mRNA, but the method is cumbersome and as of now only appears to work with fibroblasts. Piggyback may be an attractive alternative but published studies in human cells are limited and excision of the piggyback insertion has not been reported in human iPSCs. The remaining methods that have been discussed all either have severe limitations or have not been validated with the stringency required to express full confidence in their capabilities. Although there is no universal method that can handle every situation, at least one of the methods described in this chapter should be able to cover nearly every need for a researcher attempting to produce iPSCs. Part 3, Pluripotency Validation. In the first attempts at reprogramming, it was anticipated that the introduction of multiple factors would yield multiple cell fates, thus necessitating the use of a selection system whereby only cells that reactivated escape-specific genes could survive. Several criteria have been set forth to ascertain whether a fully reprogrammed state has been achieved, which include an array of unique features associated with pluripotency, encompassing morphological, molecular, and functional attributes. At a functional level, iPSCs must demonstrate the ability to differentiate into lineages from all 3 embryonic germ layers. A hierarchy of criteria has been put forth, and in order of increasing levels of stringency, these include 1. In vitro differentiation, 2. Teratoma formation, 3. Chimera contribution, 4. Germline transmission, and 5. Tetraploid complementation, direct generation of entirely escape/iPSC-derived mice. On a molecular level, iPSCs must display gene expression profiles that are indistinguishable from ESCs, which extends to the display of other associated features, including 1. Protein-level expression of key pluripotency factors, for example, OCT4, NANOG, and escape-specific surface antigens, for example, CA1 in mouse, CA3/4, TRA160/81 in human. 2. functional telomerase expression, and 3. Expression of genes involved in retroviral silencing, such as de novo methyltransferases and TRIM28. As performing all available assays for the demonstration of pluripotency is infeasible, a suggested minimal set of criteria should be fulfilled in order to ascertain that a genuine iPSC has been obtained. Accordingly, these include 1. All morphological attributes, including unlimited self-renewal. 2. Expression of key pluripotency genes with a concomitant downregulation of lineage-specific genes associated with the cell of origin. 3. Transgene independence, and 4. Proof of functional differentiation through the highest stringency test acceptable. Although many stem cell researchers dream of iPSCs being used to treat disease directly, the clinical utility of iPSCs, for the moment, is not as exciting as their role in research and their usefulness in modeling disease. iPSCs have given us the ability to look at disease in a very meaningful way, in large part because we can study the disease on an individual level, in the context of the patient's unique genetic makeup. This personalized look at disease is incredibly powerful and will certainly inform the development of new and exciting treatments in the future. Related products and services can be found at www.creativebioarray.com.